Welcome to a brand new episode of Neovirus. I'm your host Dazai, and today we'll be diving into the intricate world of a very unique human virus that often flies under the radar but has important implications for human health. Joining me today is Marzia, the newest expert on the human cytomegalovirus, or Hcmv for short.
Thank you, Dazai. I'm delighted to be here and discuss this very intriguing virus.
Before we dive in, let me remind you of this week's podcast challenge. Your task is to describe and explain Hcmv to an audience completely unfamiliar with the concept of a virus.
Ooh, that sounds like a very fun challenge.
Okay then, let's start at the molecular level. Can you give our listeners a more detailed understanding of the human cytomegalovirus, such as its morphology and genomic structure?
Certainly. So in terms of a virus, morphology refers to the structure of the virus. So basically what it looks like on the outside. Hcmv contains a host-derived envelope that contains glycoproteins. Glycoproteins are basically proteins that have a carbohydrate attached to it. And the genome is encased by an icosahedral-shaped nucleocapsid with a T of 16 that contains 162 capsomeres. Capsomeres are basically tiny subunits that make up the nucleocapsid. The nucleocapsid basically protects the genome inside the virus. Lastly, herpes virus basically contains a tegument.
Tegument. That sounds unfamiliar. Can you please define that?
Of course. So viral teguments are basically proteins that are found in the spaces between the envelope and the nucleocapsid. These clusters of proteins are unique to all herpes viruses.
Interesting. What about the genome?
Oh yes, now let's talk about the genome. So a genome refers to the entire set of genetic material present within an organism. In our case, Hcmv is characterized by 208 kilobase monopartite double-stranded DNA. This means that the entire genome is found in a straight line and in one whole part. The virus also happens to have a DNA genome and is double-stranded, much like our own human genome. So Hcmv also belongs to the virus family Herpesviridae. These viruses have some of the largest genomes amongst all of human viruses, and as such, it codes for 208 proteins. These proteins are involved in the viral life cycle, the host immune system evasion, and latency establishment.
Fascinating. I remember hearing latency establishment. Can you elaborate on that?
Oh yes. So this is one of the most interesting parts of this virus. It can essentially establish a lifelong infection in the host after initial exposure.
Lifelong infection? That sounds ominous. How does the virus manage to persist in the body for so long?
So it's actually a combination of factors. Hcmv can detect and it can infect many different types of cells, such as epithelial cells, endothelial cells, fibroblasts, and immune cells. Once someone is infected, the virus can remain latent in certain cell types, such as CD43+, as well as CD14+, meaning that it's present within the human immune cells, but not actively replicating. This ability to go dormant helps the virus evade the immune system and reactivate later when the immune system is compromised.
So it's a master at evading the body's defense system.
Exactly.
Okay then, what are some of the primary ways people contract Hcmv?
Hcmv is primarily spread through bodily fluids such as saliva, urine and feces, and breast milk. Transmission can also occur through direct contact with infected individuals or by sharing contaminated objects, blood transfusions, and organ transplants. It can also be transmitted from an infected mother to her baby during childbirth.
Let's shift gears and talk about the clinical implications of Hcmv. For most healthy individuals, is the infection symptomatic?
Interestingly, in the majority of healthy individuals, Hcmv infections are actually asymptomatic. Many people might not even realize that they have been infected. However, in certain populations such as infants, elderly, and those with a weakened immune system, the variant can cause more of a severe outcome. The virus can also target various organs, leading to conditions like pneumonia, retinitis, as well as gastrointestinal issues, and even cancer.
I see. Then what are some of the complications that are associated with Hcmv infection in these high-risk groups?
So in newborns, Hcmv infection can actually lead to a condition known as congenital cytomegalovirus, which can result in things like developmental issues, hearing loss, vision loss, and even microcephaly, among other long-term health problems. In immunocompromised individuals, such as those with HIV or those undergoing an organ transplant, Hcmv can actually cause severe and potentially life-threatening complications.
So it seems like Hcmv is a significant concern in specific vulnerable populations. Are there any effective treatments or vaccines available?
Currently, there is no specific antiviral treatments for HCMV, and a vaccine is not yet available, but management primarily involves supportive care. The main issue with the antiviral medications that did exist is that HCMV can easily develop resistance to these antiviral drugs, especially in AIDS patients and those who have previously received other antiviral treatments. However, research is ongoing to develop effective vaccines and therapies.
Great. So speaking of research, what are some of the recent breakthroughs or advancements in the study of HCMV?
The field of HCMV research has seen significant progress in recent years. Scientists are gaining a deeper understanding of the virus's molecular mechanisms, which could pave the way for the development of targeted therapies.
And could you please share a specific example?
Of course. So a recent paper published by Cmmb et al. In 2020 explored how to exploit the virus's dependency on the host's mitochondria to develop novel antivirals. So in short, when HCMV infects a cell, it changes how the cell produces energy. It makes cells increase in a process called glycolysis and oxidative phosphorylation. Glycolysis is the process of breaking down glucose into energy that happens within the cytoplasm, and oxidative phosphorylation generates ATP, which is essentially the energy currency. This process happens within the electron transport chain that is found in the inner mitochondrial membrane. To add on, the viral infection changes the shape and the function of the mitochondria. For example, the mitochondria normally undergoes something called fission and fusion, meaning they split into smaller parts and then come back together. But in cells that are infected with HCMV, these mitochondria are unable to fuse back together. The mitochondria also responds to viral infection by increasing the use of ATC, which leads to the release of something called reactive oxygen species, or ROS. And at high concentration, ROS has the potential to damage the host's DNA.
Oh wow, the mitochondria is truly powerful.
Yes, it is. And another very cool thing about the mitochondria is that it actually contains its own set of DNA that holds crucial instructions to help make energy for the cell. It essentially codes for proteins that are used within the ATC. In this specific experiment carried out by Cmmb, scientists have essentially taken out the mitochondrial DNA and found that the virus struggles to replicate within the host cell. This suggests that the virus requires the mitochondria, specifically the ATC, to replicate and survive. This also means that the number of mitochondria within the cell multiplies as HCMV induces something called mitochondrial biogenesis.
Okay, so why is this specific research important?
That's because we can exploit the virus's dependency and interactions with the mitochondria to develop potential new antiviral treatments, especially because we know that the host cell metabolism is vital to viral replication. We also happen to know that HCMV does not actually kill the host cell because it is a persistent virus, and it also happens to have a very slow replication cycle. These two factors allow for multiple potential drug targets throughout its life cycle to find a possible treatment.
That's encouraging news. Can you share some insight into the challenges researchers face when studying HCMV?
Oh, of course. So one of the main challenges is the virus's ability to establish latency, making it very difficult to completely eradicate from the body. So understanding the factors that contribute to latency and reactivation is crucial for developing effective treatments. Additionally, there is a lack of a small animal model that accurately recapitulates HCMV infections in humans, which poses a really big hurdle in preclinical studies.
That definitely sounds challenging. Now let's delve deeper into the interaction between HCMV and the immune system. How does the virus manipulate the host's immune system?
So HCMV has evolved various strategies to evade immune detection and disruption. It can modulate the expression of certain viral proteins to interfere with the host's immune signaling pathways, such as preventing natural killer cells from doing their job, as NK cells are responsible for killing virally infected cells and tumors.
It sounds like HCMV has a sophisticated strategy for dodging the immune system. Does this evasion contribute to the lifelong persistence of the virus in the host?
Absolutely. The ability to evade the immune system is a key factor in the virus's ability to establish lifelong infection. By going dormant and then reactivating, HCMV can persist in the host for an extended period of time, sometimes even for the entire life of that individual.
Interesting. As we wrap up, what do you envision for the future of HCMV research, and why should our listeners care?
The future of Hcmv research actually holds a lot of promises. So Hcmv is found within 85% of the population and can easily be transmitted as previously seen. So finding a cure or a mechanism to prevent infection to Hcmv is crucial. A cure or vaccine for Hcmv can also aid in the development of therapeutics for other herpes viruses that are also extensively affecting humans.
Thank you, Marcia, for shedding light on the intricacies of human cytomegalovirus. It's a complex subject, but your insights have certainly made it more accessible to our listeners.
My pleasure, Dazai. Thank you for having me on Nanovirus. Hopefully, I did the challenge justice.
And thank you, dear listeners, for joining us for another episode. If you have any questions or topics you'd like for us to explore in future episodes, feel free to reach out. Until next time, stay curious and stay informed. This is Dazai signing off.
Yeah!